Hello everybody, it's Mrs. Pound and we are studying Chapter 7 on Biotechnology from BJU Biology's 4th edition. We are now studying Section 7B on Practical Uses of Biotechnology. And today I want to talk about subsection 7.5 on DNA fingerprinting. So our objectives today will be to define DNA fingerprinting and describe the use of DNA fingerprint technology. So what is a DNA fingerprint? It's a distinctive pattern of bands composed of fragments of an individual's DNA, and you can see it in the background. These are DNA fingerprints. Only about 0.1% of the DNA is different between individuals. So when we do a DNA fingerprint, we're not looking at a person's entire genome. We're looking at a very small percentage of that. And the reason most of our genome is the same is because we are coding for, in general, most of the same proteins. The only differences would be, say, if there is a genetic disease, that would show up differently between individuals, eye color, hair color, those sorts of things. But for the most part, we are making a lot of the same proteins. So what can DNA fingerprinting be used for? It can be, first of all, used for pursuing justice. If there is a crime done and there is blood left at the scene or hairs, some kind of sample that we can extract DNA from, then we can do a fingerprint to help us narrow the suspects and find out who actually is guilty. It can also be used, if we can find a DNA sample, to release somebody from prison if they have been wrongfully imprisoned. They have used DNA samples to do that, where people were found guilty who actually were not. It's also used in diagnosing human genetic disorders. Some disorders that it can be used to diagnose currently are cystic fibrosis, hemophilia, sickle cell disease, Huntington disease, and some types of Alzheimer's disease. So what does DNA fingerprinting involve? We're going to be doing a lab in class that kind of simulates going through this process. So the first thing you have to do is you have to isolate a sample, usually a blood sample or a hair sample. Those are commonly used. A lot of times the samples are too small, so we have to use something called polymerase chain reaction, PCR. And what polymerase chain reaction does is it makes millions of copies of DNA in a short period of time. So it multiplies the DNA so we have a large enough sample to look at. This actually, PCR is what is also used for some of the testing for COVID-19 currently. Is they take a sample and it's too small, so they put it through polymerase chain reaction in order to multiply the DNA. Step 2 is the use of restriction enzymes. They then cut the DNA up into smaller pieces at certain spots. Step 3 is gel electrophoresis. Gel electrophoresis sorts the DNA fragments by size, so you see the equipment in the background, and electrical charge is put on this equipment, and the DNA fragments travel. The longer the fragment, the slower it travels, and the shorter fragments travel faster. Step 4 is the transfer of fragments from gel to nylon sheet so that it can be read. 5 is the addition of radioactive probes so that the lines can be seen. And 6 is you finally have your DNA fingerprint, which you can see in the background, and you can compare the DNA to see if it's a match for, say, a disease, because the lines would be in different places for an individual who does not have the disease and who does have the disease. So what they would do is they would run a gel, they would have your sample, and then they would have some control samples. They would have a normal individual's DNA and an individual who has the disease you're looking at, and they would run those and see how your DNA compares to the two controls. So our objectives today were to define DNA fingerprint and describe the uses of DNA fingerprint technology. Don't forget your 5 questions in your margin, and remember to answer the questions at the end.